In 2007, a team of archaeologists climbed into a limestone cave in the northern Philippines on an island called Luzon. The cave was called Callao, and it was miserable to reach. You needed ropes, headlamps, and a willingness to crawl through narrow, pitch-black passages just to get to the dig site. For decades, mainstream archaeology had largely written the Philippines off as a dead end for human origins research, a scattering of tropical islands too remote, humid, and too poorly preserved to hold anything as significant as what kept turning up in Africa or Europe. Most researchers had ignored Callao Cave for exactly that reason: too difficult, too remote, and, as far as anyone assumed, too unlikely to hold anything worth the risk. But in 2007, a team from the University of the Philippines went in anyway, and what they pulled out of the cave floor would take 12 years, 2 more expeditions, and a small army of scientists to even begin to understand. At first, it looked ordinary: stone tools, animal bones, the usual scraps left behind by ancient people. Then someone found the teeth. 3 molars, worn down from decades of chewing, small and strange, unlike anything found anywhere else in the region. Carbon dating placed them at 67,000 years old, making them the oldest human remains ever discovered in the Philippines. For over a decade, those teeth sat in storage while scientists argued about what they meant. An initial analysis in 2010 tentatively described their remains as belonging to the genus Homo without committing to a species because there simply was not enough material to say more. Were they from modern humans who had arrived earlier than anyone expected? Or from something else entirely, a population nobody had a name for yet? Nobody could agree because 3 molars are not enough evidence to overturn a species. And the team knew that publishing too soon on too little material would invite exactly the kind of skepticism that has sunk other extraordinary claims in paleoanthropology. So they waited, and they kept digging. Then, in 2019, everything changed. The team went back into Callao Cave and found more: a femur, finger bones, toe bones, another set of teeth. And when they finally pieced it all together, the bones did not belong to Homo sapiens. They did not belong to Neanderthals. They did not match Denisovans either. They belonged to something new, a species science had never documented before, and it was about to rewrite the timeline of human evolution in Southeast Asia. If you are enjoying this dive into ancient human origins, hit subscribe, because I cover discoveries like this one every single week. The researchers named the new species Homo Luzonensis, after the island where it was found. And almost immediately, the bones started raising questions nobody could answer. The teeth were small and simple, closer in shape to modern humans than to any archaic hominin. But the finger and toe bones were sharply curved, a trait normally associated with species that spent a significant part of their lives climbing trees, features last seen in hominins that went extinct roughly 2 million years ago. The femur, meanwhile, was thick and robust, built for a body that walked long distances on the ground. It was as if someone had assembled a hominin out of spare parts from 3 different eras of evolution. Paleoanthropologists studying the fossils described the combination as unlike anything they had encountered before, a mosaic of primitive and modern traits appearing together in a single, small population isolated on a Pacific island. One of the foot bones in particular drew intense scrutiny. Its curvature closely resembled that of Australopithecines, the famous group of ancient hominins that includes Lucy, who walked across East Africa roughly 3 million years earlier. And it was strikingly similar in shape and length to a foot bone belonging to Homo floresiensis, the so-called hobbit species, already known from the nearby island of Flores. Meanwhile, one of the primolars recovered from Callao Cave had 3 separate roots, an oddity rarely seen in modern human teeth. With only 13 bones and teeth recovered from at least 3 individuals, 2 adults and a child, there was not enough material to say how tall Homo luzonensis stood, how it lived day to day, or even exactly where it fit on the human family tree. Some researchers argued it might be a late surviving offshoot of Homo erectus, the first hominin species known to have left Africa. Others pointed to the primitive foot and hand bones and wondered if Homo luzonensis descended from something far older, a lineage that left Africa long before erectus and evolved in total isolation ever since. The debate remains unresolved, and it might stay that way, because there was one obvious way to settle it once and for all. If scientists could recover ancient DNA from the bones, they could compare Homo luzonensis directly to every other known human lineage and finally place it correctly on the tree. So that is exactly what they tried to do. It did not work. Extracting genetic material from bones tens of thousands of years old is difficult, even in ideal conditions. From the moment an organism dies, enzymes inside its own cells begin breaking down its DNA, while microbes in the surrounding soil move in and accelerate the process further. What little genetic material survives that initial assault keeps fragmenting for thousands of years afterward, shattering into shorter and shorter strands. until what is left resembles a book that has been shredded, scattered, and left out in the weather. Sequencing that kind of degraded material requires piecing together millions of tiny overlapping fragments, and then filtering out the overwhelming background noise of bacterial and fungal DNA that has colonized the bone in the meantime. Ideal conditions for preserving that fragile genetic material are cold and dry, which is why almost every major ancient DNA breakthrough of the last 2 decades, Neanderthal genomes, Denisovan genomes, the reconstructed migration routes across ancient Europe, has come out of caves in Siberia and Europe, where freezing temperatures act like a natural deep freezer, slowing decay to a crawl. The Philippines offered the opposite of ideal conditions. Callao Cave sits in a tropical environment, hot and humid year-round, and heat, combined with humidity, destroys DNA far faster than cold ever could, breaking down the chemical bonds that hold genetic material together within a fraction of the time it would take in a Siberian permafrost. When the research team analyzed the Homo Luzonensis remains, they found exactly what tropical preservation predicts: DNA that was too degraded, too fragmented, and too contaminated to sequence into anything usable. The attempt failed, and with it, the most direct way to answer the biggest question about this new species, where it actually came from and how it relates to every other hominin ever discovered, went dark. That should have been the end of the story, another fascinating fossil discovery destined to remain a mystery forever, filed away next to dozens of other tantalizing but genetically silent finds. Except scientists realized there was a second place to look, and it was not in a cave at all. It was in the blood of people living in the Philippines today. Here is the logic. If Homo luzonensis, or a population closely related to it, survived on Luzon until relatively recently, and if modern humans eventually arrived and encountered them, there's a real chance the two populations interbred. When that happens between human lineages, the genetic evidence does not stay locked in old bones. It gets copied forward, generation after generation, embedded in the DNA of the descendants. You do not need the original skeleton if the genetic signature is still walking around, diluted but present, in someone alive, right now. So researchers turned their attention to the genomes of modern populations in the Philippines, and in particular to groups known as Negritos, indigenous communities with some of the oldest continuous ancestry in the islands. In 2021, a team led by researchers from Uppsala University in Sweden, working in partnership with local universities and indigenous communities across the country, published the results of an enormous genetic survey. Roughly 2.3 million genetic markers collected from more than 1,000 individuals across 118 distinct ethnic groups throughout the Philippines, including 25 different self-identified Negrito populations. They combined that dataset with high-coverage genomes sequenced directly from Ayta Magbukon individuals and from Australopapuan populations for comparison. What they found was staggering. One group in particular, the Ayta Magbukon of Luzon, carried the highest level of Denisovan ancestry ever recorded in a living human population, anywhere on Earth. Not the highest in Southeast Asia. The highest in the world, roughly 30 to 40% greater than Papuan Highlanders, who had previously held that record by a wide margin. Sit with that for a moment. Denisovans are an archaic human population first identified not from a full skeleton, but from a single small finger bone found in a Siberian cave in 2010, a discovery so slight that almost nobody expected it to reshape the entire field of human evolution. Their DNA shows up today in people across Asia, the Pacific, and Australia, but always in small, scattered traces. The idea that a group living on a small island in the Philippines could carry more Denisovan ancestry than anyone else on the planet did not fit the existing map of human migration at all. The researchers concluded that this could not simply be leftover DNA carried in from the mainland by later migrants. The pattern only made sense if there had been a separate, independent interbreeding event, Denisovans, or a very closely related population, meeting the ancestors of the Ayta directly on or near the Philippine Islands themselves, entirely apart from the mixing events that produced the Denisovan ancestry seen in New Guinea and Australia. In other words, there was another ghost population living in the Philippines, one that left no bones we have found yet, but left an unmistakable genetic fingerprint in people alive today. Some researchers now wonder whether that ghost population and Homo luzonensis are connected, whether the small hominin from Callao Cave and the mystery lineage detected in Ayta genomes are describing the same vanished people from two completely different kinds of evidence, one skeletal, one genetic. Nobody can prove that link yet. But the coincidence, a distinct archaic human presence identified by bones on Luzon, and a distinct archaic genetic signal identified independently in the people who live there now, is exactly the kind of overlap that keeps scientists awake at night. To understand how any of this was even possible, you need to understand what the map of Southeast Asia looked like during the Ice Ages. Today, the region is a scattering of islands, the Philippines, Borneo, Java, Sumatra, separated by stretches of open ocean. But that is a very recent arrangement. During the last glacial period, so much of the planet's water was locked up in ice sheets that global sea levels dropped by well over 300 feet. The shallow seas separating those islands from mainland Asia simply were not there. In their place was dry land, an enormous landmass known to scientists as Sundaland, roughly twice the size of India, connecting Thailand, Malaysia, Sumatra, Java, and Borneo into a single continuous continent. Rivers cut through valleys that are underwater today. Forests covered plains that are now the floor of the Java Sea. Elephants, rhinoceroses, tigers, and early humans moved across that landscape without ever encountering an ocean. Sundaland functioned as a migration corridor, and every hominin species that ever reached the western part of island Southeast Asia almost certainly walked there, following rivers and coastlines, long before anyone needed a boat. But Sundaland only got you so far. Further east lies an invisible boundary biologists call the Wallace Line, named after the naturalist Alfred Russel Wallace, marking a deep water channel that never closed even at the height of the ice ages. Beyond that line sits a region known as Wallacea, which includes Sulawesi, Flores, and the Philippines, islands that were never connected to the mainland by dry land, no matter how far sea levels dropped. Reaching them required crossing open water, whether by accident, by drifting on natural rafts of vegetation during storms, or through some form of deliberate seafaring we still do not fully understand. The fact that hominin populations, and their stone tools, show up on the far side of that line at all, on Flores, on Sulawesi, on Luzon, tells us that whoever these early humans were, they were capable of doing something scientists once assumed only anatomically modern humans could pull off. But Sundaland was not permanent. Roughly every 100,000 years, the climate cycled between glacial and interglacial periods, and every time the ice melted and sea levels rose again, Sundaland began to drown. Populations that had spread freely across the continent suddenly found themselves stranded on shrinking pockets of land as rising water filled in the lowlands around them. Some of those populations died out. Others adapted to isolation, and over enough generations, isolation reshaped them into something new. This cycle repeated itself again and again, and different hominin species arrived on different islands during different climate windows, each cut off at a different point in time. Homo erectus reached Java over 1 million years ago, during one of the earliest and most extreme drops in sea level, and lived there in isolation for hundreds of thousands of years, evolving distinct traits along the way. On the neighboring island of Flores, another population, either descended from Homo erectus or from an even older lineage that made the same crossing, shrank dramatically over time in a process called island dwarfism, eventually becoming the species discovered in 2003 and nicknamed the Hobbit, Homo floresiensis. Standing barely 3.5 feet tall with a brain roughly the size of a chimpanzee's, Homo floresiensis nonetheless made sophisticated stone tools, hunted dwarf elephants, and controlled fire, surviving on Flores until perhaps as recently as 50,000 years ago, meaning its existence overlapped in time with both Homo luzonensis on Luzon and the earliest modern humans pushing into the region. Recent excavations on the neighboring island of Sulawesi have pushed the presence of tool-making hominins back even further still. In 2025, researchers working at a site called Callao uncovered stone tools dated to somewhere between 1 million and 1.5 million years old, nearly 1 million years earlier than any previously known evidence of hominin activity on that island, alongside fossilized remains of an extinct pig species that helped anchor the dating. No hominin bones turned up alongside those tools, which means whoever made them remains completely unidentified. But the discovery confirms that ancient hominins were crossing open water, an island hopping through Wallacea, far earlier and far more successfully than anyone assumed even a decade ago. By the time anatomically modern humans arrived in island Southeast Asia, somewhere around 50,000 years ago, they were not walking into an empty landscape. They were walking into a region already inhabited by multiple distinct hominin populations, each shaped by tens of thousands of years of isolation on a different island, each carrying its own unique combination of physical traits, and as we now know, its own distinct genetic signature. And when modern humans encountered these populations, in at least some cases, they did not simply replace them, they interbred with them. Denisovan ancestry itself was first identified through fossils from Denisova Cave in Siberia, but the genetic story of Denisovans turned out to be far bigger and far stranger than a single cave in one location. Papua New Guinea and Aboriginal Australia carry some of the highest levels of Denisovan ancestry outside the Philippines, and researchers studying those genomes have found evidence suggesting not one, but multiple separate waves of interbreeding with distinct, deeply diverged Denisovan-related populations, essentially several different ghost lineages layered on top of each other across the region, some more closely related to the original Siberian Denisovan individual than others, implying a whole family of related archaic populations scattered across Asia and the Pacific, rather than one uniform group. That means the story of ancient human interbreeding in Southeast Asia and the Pacific was not one clean event. It was many events, happening in different places at different times, with populations that may have looked and lived very differently from one another, depending on how long they had been isolated and on which island they called home, all filtered down into the DNA of people alive today, sometimes in amounts high enough to be detected easily, and sometimes in traces so faint they only show up when researchers specifically go looking for them. And this archaic ancestry was not simply passed on as inert genetic baggage. In several documented cases, it provided real, measurable survival advantages. A variant of a gene called EPAS1, which helps the body use oxygen more efficiently at extreme altitude, is common among Tibetans today and traces directly back to Denisovan ancestry, giving highland populations a biological edge against altitude sickness that lowland populations simply do not have. Immune system genes inherited from Denisovans and Neanderthals have also been shown to broaden the range of pathogens certain modern populations can recognize and fight off, particularly genes tied to the human leukocyte antigen system, which helps the immune system distinguish the body's own cells from invading pathogens. Researchers have argued that these inherited variants gave early modern human populations, moving into unfamiliar environments, a head start against local diseases their African-adapted immune systems had never encountered before, a genetic toolkit assembled not from scratch but borrowed from species that had already spent hundreds of thousands of years adapting to their environments before modern humans ever arrived. Some scientists studying Southeast Asian and Melanesian populations have also floated the possibility that archaic ancestry contributed to variants involved in metabolism and energy storage, potentially useful in tropical environments with seasonal food scarcity, though this particular line of research is still preliminary and far less settled than the immune and altitude findings. When you consider that some of the archaic populations in island Southeast Asia had potentially been isolated there for tens of thousands or even hundreds of thousands of years, it starts to make sense that their genes carried adaptations useful for surviving specifically in that environment. Adaptations that modern humans, arriving fresh out of Africa with genetics tuned for a completely different climate, could benefit enormously from inheriting, which raises an obvious and uncomfortable question. If interbreeding conferred real advantages, why did every one of these archaic populations disappear? Why is Homo sapiens the only human species left standing? Honestly, nobody knows for certain. There are competing theories. Some researchers argue that modern humans had more complex social networks and cooperative behavior that allowed larger, more resilient populations to simply outsurvive smaller, more isolated archaic groups during periods of environmental stress. Others suggest that modern humans carried diseases that archaic populations had never been exposed to and had no immunity against. A grim preview of what would happen again thousands of years later when different human populations made contact around the globe. Some scientists have proposed that hybrid offspring between modern humans and archaic populations may have faced fertility problems in later generations, a slow genetic drag that could have caused smaller archaic populations to be gradually absorbed rather than replaced outright, a process that would leave behind exactly the pattern we see today. Isolated fragments of archaic DNA scattered through modern genomes with no surviving pure-blooded population left to point to. It is also worth remembering that these were often extremely small populations to begin with, isolated on single islands for tens of thousands of years, which on its own makes any group far more vulnerable to a single bad drought, a disease outbreak, or a volcanic eruption than a large, connected population spread across a continent would ever be. And then there's a more unsettling possibility, one that some researchers take seriously. Maybe these populations never really went extinct at all. Maybe calling them extinct is simply the wrong word for what happened. If a population's distinct skeleton disappears from the fossil record, but between 5 and 10%, or in the case of the Ayta Magbukon, an even larger share of a living population's genome still traces back to that lineage, is that population truly gone? Or is it simply distributed, no longer concentrated in one place, one body, one skeleton, but spread across the cells of millions of descendants who have no idea they are carrying it? What we know for certain, and this is really the headline that all this evidence points toward, is that modern humans are not a pure single lineage, tracing back cleanly to one African origin point with no detours along the way. We are a genetic patchwork. Nearly every population outside sub-Saharan Africa carries measurable DNA from at least one, and in many cases several, archaic human populations that are technically supposed to be extinct. Southeast Asia, and the Philippines specifically, appears to be one of the most intense zones of that mixing anywhere on the planet. A place where at least 4 distinct hominin lineages, Homo erectus descendants, Homo floresiensis, Homo luzonensis, and one or more Denisovan-related populations may have coexisted at overlapping points in time before eventually folding into the single species that exists today. And this is very likely not the end of the story. There are still enormous numbers of unexcavated caves scattered across the limestone landscapes of the Philippines, Indonesia, and the wider Wallacean region, any one of which could contain more bones, more teeth, more fragments of a story we have only begun to piece together. The 2225 discovery of stone tools on Sulawesi dating back well over 1 million years shows that hominins were crossing these ocean barriers, deliberately or by accident, far earlier and far more frequently than researchers assumed even a few years ago. And in most of these cases, we still have no fossils at all, only tools, meaning the identity of the tool makers themselves remains completely unknown. Every new excavation season carries the possibility of finally recovering bones with genetic material intact, or finding an entirely new population nobody has documented yet. And every new genome sequenced from a living population in the region carries the possibility of revealing yet another hidden ghost lineage, another archaic population that left no fossils we have found, but left its mark quietly, invisibly, in blood and cells and DNA passed down across thousands of generations. Researchers involved in the Ayta genetic study have already said publicly that sequencing more genomes from more populations in the years ahead should reveal further insight into exactly how much archaic inheritance has shaped human biology and adaptation across the region, which suggests this is very much an ongoing investigation rather than a closed case. For most of the last century, the story of human origins was told as something simple and linear. Modern humans evolved in Africa, left the continent, and spread across the world, eventually becoming the only human species left standing, a single triumphant line stretching in an unbroken path from an African cradle to everywhere else. The genetic and fossil evidence emerging from places like Callao Cave, Flores, and the genomes of the Ayta Magbukon tells a far messier, far more interesting story. We did not simply replace the archaic populations we encountered. In at least some cases, we absorbed them, took their genes, and in the process took traits that had taken those populations tens of thousands of years to evolve in some of the harshest and most isolated environments on the planet. The bones sitting in Callao Cave for 50,000 years could not give up their DNA. The tropical heat and humidity made sure of that. But the story did not need the bones to survive. It survived instead in the genomes of the people living across those same islands today, carried forward silently through generation after generation, waiting for scientists with the right tools to finally notice it was there. The real question now is not whether interbreeding with these lost populations happened. The evidence for that is no longer in serious doubt. The real question is how many more of these ghost lineages are still hidden in the DNA of people alive right now, waiting quietly in genomes nobody has thought to sequence yet, and what else they might still be able to teach us about our immune systems, our metabolism, our history, and about who we actually are.